Good morning, ma'am, sir, and all my friends. Today, me and my friends will talk about human and animal cells in pharmacy and human disease with myself, Angela Mervin, Dinia Octaviani, and Fatima Azara Sarwinata. So, this is the outline of what are we going to talk about. The first is about the organelles, humans and animal cells, the application of cell theory, types of cells, and disease in human cells. First, we're gonna talk about the human and animal cells. Now, I'm gonna talk about the nucleus and nucleus. Nucleus only found in eukaryotic cells, and it describes DNA with the proteins, while nucleus is the site of ribosome and ribosomal RNA production. Now I'm going to talk about the endoplasmic reticulum. It's particularly for synthesis, folding, modification, and transport of proteins. There are also two kinds of endoplasmic reticulum. It was the RAF-RA. It's named for RAF because the ribosome is attached to its outer, while there are also smooth end, which is not associated with ribosomes, and the for RAF-RA is to synthesis of protein, while the smooth RA synthesizes lipids. Golgi apparatus which a warehouse of the cell, sorting, shipping, and even some manufacturing. And it also provides vesicles as a transport for molecules to other part of the cells. Next, it was mitochondria. The function of mitochondria is to ATP and also for the cell respiration. The next is ribosomes, cell structures that makes protein, but what the location of ribosome in a cell determines what kind of protein it makes. Next is about the cytoplasm. It's a substance of life, and it's also have some three kinds of the main components. The first one is cytosol. It consists of cytoskeleton, filaments, organic molecules, salt, and water. And the second is organelles, and third is various cytoplasmic. The function of cytoplasm itself is to be the site for most of the enzymatic reaction, and also the site where all of the cells are expanded and grow. Next, my friend Dinia will continue our presentation.
Thank you, Marfin. Now I'm going to be talking about centrioles, lysosome, and peroxisome. First, centrioles are organelles which are only active during cell division. They produce spindle fibers which attaches to chromosomes, and these fibers pull a copy of each chromosomes to opposite sides of the cells so that each daughter cells contains new DNA needs. Centrioles are also involved in the formation of flagella and cilia, which is also involved in cell movement. Lysosome is an organelle which contains digestive enzymes such as glycosidases, proteases, and sulfases, and this means that they're low in pH because it's used to break down excess or worn out cell parts. Peroxisomes are found in all eukaryotic cells, and its function is to break down organic molecules by the process of oxidation to produce H2O2, also known as hydrogen peroxide. It converts this to oxygen in water. And examples of oxidative enzymes include catalases. And the difference between peroxisome and lysosome is that lysosome break down biological polymers, for example, proteins and polysaccharides, whereas peroxisome oxidize organic compounds. Next, I'm going to be talking about vacuoles. So unlike plant cells, vacuoles and animal cells are actually used for spaces for storage and waste products. It also involves itself in endocytosis and exocytosis, which are fundamental to the process of intracellular digestion. Food particles are taken into the cell via endocytosis into a vacuole, whereas the leftover waste product of digestion are carried to the plasma membrane by the vacuole and eliminated through the process of exocytosis. Next, cytoskeleton maintains cell shape, anchors organelles, and assists with transport. Cytoskeletons are composed of three things: microfilaments, microtubules, and intermediate filaments. Microfilaments assist with cell movement and are made out of a protein called actin. Actin works with another protein called mitosin to produce muscle movements, whereas microtubules are cylindrical tubules, which are subunits of protein tubules, and intermediate filaments are used to provide tensile strength.
Now we will talk about the similarities and differences of the human cell and animal cell.
Well, actually, there is not much of a difference between the human cell and the animal cell because humans are classified as animals. But in this case, we are looking at the point of view where the human cells are the various types of cells that makes up the human body. And an animal cell is a type of eukaryotic cell in which we have discussed most of the organelles and functions previously with Dinya and Mervin. Now, as you can see in the slide, there is the similarities and the differences. The similarities of the human and animal cells are that they do not have a cell wall. They do not have chloroplast or a large vacuole, such as a plant cell would have. Second is that they have a cell membrane, a cytoplasm, and other organelles that were mentioned previously. And then third, they form multicellular organisms. Now, the differences would be as follows. Well, like I mentioned earlier, in terms of definitions, we refer to the animal cell as a eukaryotic cell. And as for humans, we define them as a functional unit of the human body. In other words, the various types of cells that makes up the human body. Number two, in an animal cell, the different size of genomes exists based on the species. As for human cells, they have a genome with 3 billion base pairs. And number three, just like the size of genomes, animal cells' numbers of protein coding genes depend on the species. As for human cell, they may have around 25,000 protein coding genes. Moving on about the application cell theory. Why is it important for us to know the different organelles and the functions of the organelles as well as the similarities and differences of the human and animal cell? In pharmacy specifically, you may ask. Well, there are many reasons as to why it is important, but the two main reasons that I would like to point out are to understand the mechanism of drugs at a cellular level. There are many reasons as to why it is important for us to know this. But the two main reasons that I would like to point out is that one of them is to understand the mechanism of drugs at a cellular level. In the field of pharmacy, we would be dealing with drugs frequently, right? Therefore, it is crucial for us to be knowledgeable about the several aspects that comes with a drug, or the several aspects of a drug, such as the mechanism of the drug and how it works. And knowing it at a cellular level is that we can see how the cells in our body would respond to the drug that we take and see if it would affect it in a good way or a bad way. And I feel like that is really important because we want to ensure that it will affect it in a good way. The second reason I would like to point out is the diagnosis of diseases. There are various types of diseases in this world, and as pharmacists, it is important, to come up with a treatment or a cure for it. And knowing the information we spoke about previously can help us identify the cause of these diseases and enable us to make the correct treatment for it. It is known that one of the causes of diseases could be from a damaged cell. Therefore, to understand this in a deeper sense, we will talk about the several types of cells, specifically the epithelial cell, the nerve cell, the muscle cells, and the endothelial cells. Now, I'll pass this on to Mervin, who will start us off by explaining about the epithelial cells and the nerve cells.
Thank you, Zara. Next, I will talk about four types of cells. The first one is epithelial cells, nerve cells, muscle cells, and endothelial cells. First is about epithelial cells. These cells cover organs and body cavities and have sensors called receptors. And there are also two types of epithelial cells. The first one is simple squamous. This kind of shape is flat and only have one layer. We can find it in capillary endothelium and Bowman's capsule of the kidneys, pericardium, and the peritoneum, and also pleural cavity. Second is simple columnar cells. This shape is long and only have one layer. We can find it in digestive tract and our female reproductive. The third is simple cuboidal cells. These shapes are cubes and only have one layer. You can find it in the lining of secretory ducts in the kidneys or gland. The fourth is stratified squamous cells, which is flat and arranged in multiple layers. You can find it also in pharynx, esophagus, oral cavity, uterine cervix, vagina, and skin. The fifth is stratified tuboidal cells, which is cubes and arranged in multiple layers, but you can find them in the ducts of sweat glands. The sixth is stratified columnar cells, which is rectangular and have one layer. You can find them in trachea and upper respiratory tract. And the last is stratified columnar cells, which is rectangular and arranged in multiple layers. But you can find it in expiratory duct, in the eyes, uterus, and anus. Next is the nerve cells. It's also called as neurons. The anatomy itself is cell body, dendrites, axon, myelin, cells, nodes of Ranvier, and axon terminal bundle. There are three types of neuron. The first one is sensory neurons. It makes us feel, smell, sound, sight, and hear, because these are type of neurons that activated by external physical or chemical stimulation. And the second one is motor neurons, are the types of neurons in the spinal cord that connects the organs, muscles, and different types of glands. And the last is intermediate neurons, which is connect the motor neurons to the sensory neurons, thus allowing for signals to be transmitted between the two. Next, my friend Zara will continue our presentation.
Thank you, Mervin. Now, let's move on to the muscle cells. The muscle cells are commonly known as myocytes, and they are the smallest subunits of our muscular tissue and organs in the body. The main function of the muscle cells would obviously be contractibility, right? And how this works is by the help of known proteins such as actin and myosin. The way they do this is by forming thin and thick filaments which will overlap each other to contract the smallest units of the muscle, which is known as sarcomeres. And these sarcomeres make up the myofibrils. And surrounding the myofibril, we have something called the sarcoplasmic reticulum. The role of the sarcoplasmic reticulum is to determine whether a muscle contraction will occur or not. And the way this works is that they would be activated by a brain sending impulses down to the nerve, and the nerve impulses will travel down the nerve cell to the neuromuscular junction, where a nerve cell would meet a muscle cell. In addition to that, we have three types of muscle cells, which are skeletal muscle, smooth muscle, and cardiac muscle. In skeletal muscle, they are responsible for the voluntary contractions in our body, such as the biceps, the hamstrings, and because of this, they require a lot of energy, which means they contain a lot of mitochondria. As for the smooth muscles, they are responsible for the involuntary contractions, such as the blood vessel, the respiratory tract, and in this case, unlike the skeletal muscle, they contract more slower and relax. And lastly, we have the cardiac muscles, and they are like smooth muscles in terms of what contractions they do, which is involuntary. And their key role is to generate enough contractile force so that the heart can beat effectively. This way, they can work together in unison, causing enough pressure for the blood to go around the body. That is all about the muscle cells for now. Next will be about endothelial cells, and Dinya will talk about it. Endothelium cells form a single layer that lines the blood vessel around the body, for example, the coronary artery. When endothelial cells get damaged, they can cause the development of new diseases, for example, atherosclerosis, which I'm going to be discussing in the next slides. There's a difference between endothelial and epithelial cell, one of which endothelial cells cover the inner surface of blood vessel, whereas epithelial cells, as mentioned before, cover the inner surface of internal organs and outer surface of the human body, for example, underneath the skin. Here are the main functions of endothelial cells. The first one is barrier function. The name suggests itself that barrier function is the barrier between the blood vessel and the external environment. Second one is regulation of blood flow, wound healing, prevention of thrombosis. Thrombosis in this definition means prevention of blood clots. Endothelial cells could be classed as three different things. The first one is continuous endothelium. This can be found in lung, muscle, and central nervous system. They form continuous sheet around luminous vessels, with only the gaps being adjacent to the endothelium cells. Then there's the fenestrated endothelium, and these are usually found in kidneys, the lives of intestines. These are usually more permeable than continuous endothelium. The last one is discontinuous endothelium. They can be found in blood vessels of liver and bone marrow, and they have greater permeability than both fenestrated and continuous endothelium.
Now we will talk about what kind of disease that will occur in human cells. The first is dementia. Dementia, which is caused by damage in nerve cell because it lost its ability to communicate normally, so all of the behavior, thinking, and feeling will be affected. We also call this kind of disease as lost memory, and Alzheimer is one of the most common cause of dementia. Unfortunately, there is no test to determine this disease, and it also has no cure for this, but there are some drug treatments that will help you temporarily to improve the symptoms. There are also some other that caused by the nerve cells. It was Parkinson's disease, which is a progressive nervous system disorder that affects movement, peripheral neuropathy as a result of damage to the nerve outside of the brain and spinal cord. Huntington's disease is rare, but it's inherited disease that causes the progressive breakdown of nerve cells in the brain. And the last is multiple sclerosis, MS, because the immune system attacks the protective sheet, the myelin, in the nerve cell.
Now, for this disease, it is caused by when an epithelial cell gets damaged, and specifically the stratified squamous cells type, which Mervin had mentioned before. And this disease, maybe some of you may have heard it or not, but it's called eczema, which is commonly known as atopic dermatitis. What is eczema, you may ask? Well, eczema is a skin condition that causes an itchy, red, and irritated skin. They usually begin in childhood and become better or improved as you go into adulthood. But for some people, this is not the case. They usually have chronic eczema, and this is where the eczema could last for a lifetime with occasional flare-ups. There are many triggers for a flare-up to happen, and these triggers could be soaps, environmental factors such as winter maybe, because the coldness can make our skin really dry, and allergens. And these allergens could be pollen, mold, or dust mites. Moreover, into how a damaged epithelial cell could cause eczema is that epithelial cells makes up the epidermis, which is the outermost layer of our skin. And when an epithelial cell dysfunctions, this means that there is a unregulated leakage of materials across the barrier, and this will weaken our epidermis. Therefore, the epidermis will not be able to carry out its jobs as normal, and that job is to keep foreign substances out, such as bacteria, viruses, and allergens, which I have mentioned previously is a common trigger of flare-ups in eczema. If you or someone you know feels like they have eczema, you could go to the doctors and get it diagnosed. There are many ways this could be diagnosed, such as the doctor will examine your skin, and then we also have the reviewing the patient's medical history records, and then we also have another way, which is patch testing. As for now, unfortunately, there has not been a cure for eczema, but there are a countless of ways you can treat it and care for it, such as medications. Another form of treatment is therapies. Lastly, is to try to avoid the triggers that can cause your flare-ups. That is all from me about eczema. Now I will pass this on to Dinia, who will talk to you all about atherosclerosis. Thank you, Zahra. Now I'm going to be talking about atherosclerosis. Atherosclerosis occurs when the blood vessels that carry oxygen and nutrients from your heart to the rest of your bodies become thick and stiffened, usually with plaque. For example, the coronary artery, which supplies oxygen to your heart muscles, become deprived of oxygen once there's a blockage. So the artery is usually lined with endothelium cells, as I've mentioned before, and its job is to secrete proteins to prevent the clotting of the artery. How does the endothelium cell get damaged in the first place? Here are the main triggers. The first one being high blood pressure. For example, two is too many lipid cholesterols. These are the bad cholesterols, LDLs, and toxins, for example, cigarette toxins. Once the endothelium cells get damaged, the process of atherosclerosis can start. Atherosclerosis can happen. So here in yellow, we have the LDL cholesterol and the toxins, along with high blood pressure. This can cause the damage of endothelial cells, and thus the endothelial cells get damaged. The LDL cholesterol, which accumulate in the bloodstream, start to accumulate on the damaged endothelial cells. And once they already implant themselves on the damaged endothelial cells along with the toxins, they start to oxidize, and these send signals to immune system and brings the monocytes. The monocytes then implant itself into the damaged endothelial cell, and it becomes a macrophage and it starts eating away the cholesterol. Eventually, the macrophage itself starts to gorge and die and become foam cells. These foam cells, remember that these foam cells are white blood cells. They release cytokines, which is signals which call for reinforcements. This whole process repeats whilst LDL is still being processed. While LDL is still being deposited. Then the smooth muscle cell secretes fibrous protein cap, which shields plaque from the bloodstream because it doesn't want the plaque to mix in with the bloodstream. The foam cells releases cytokines, which induces the smooth muscle cells to deposit calcium into the plaque, making it harder. The blood vessel then bulges and it restricts blood flow, and the calcium hardens the plaque, causes the blockage of artery. Once it starts to become big like this, it sometimes bursts and the thrombogenic properties start to react with the blood and it starts to harden. That's it for our presentation. Thank you so much for listening and hope to see you soon.